Hi guys, welcome back to our podcast. Today we're going to be talking about lollipops that can detect cancer. I'm Shanti.
I'm Serena.
I'm Sharvi.
And I'm Ellen. Basically, these lollipops that can detect cancer were created and researched by researchers at the University of Birmingham in the UK. Their goals are to make a less invasive alternative that could make a huge difference to patients, and they want to create a flavored lollipop test designed to spot mouth cancer early. This was co-funded by the Cancer Research UK and Swindon-based UK Research and Innovation, aka the UKRI. A team in the School of Chemistry, ran by Ruchi Gupta, has created smart hydrogels that can capture linked proteins from saliva. The basic process is that a patient would suck on a lollipop and the hydrogel would absorb the saliva, then it would be taken to a lab and the gel would be exposed to a specific color of light which would release trapped proteins for analysis. More specifically, this hydrogel is made out of precise measures of water and permeable polymer, which would isolate tiny proteins from the liquid samples. And then the certain color of lights is the only way scientists can extract the different types of proteins for testing. The goal is for it to be a more accurate test that is reliable and would take no more than 10 minutes in a clinic. Not only oral cancer, the technology could also apply further. This is because the hydrogel used for these lollipops are designed to capture specific proteins which can be adapted for other diagnostic uses, such as urine tests on different diseases. And finally, as of now, it is shown that hydrogels can only pick up proteins up to 12 hours, meaning it is not suitable for patients yet, but there's more research going on to fix this and make a better solution. Now, Shanti and Sharvi will explain more about the process more in-depthly.
Before I explain the process, I just want to talk about why early detection is really important. So many people don't find out they have oral cancer until it has spread. So late diagnosis can mean surgery, chemotherapy, or radiation, and just like in general, difficulty eating, speaking, or even breathing. So if doctors can detect cancer earlier, treatment will be less intense and survival rates will be much higher. So this lollipop could help doctors screen people who are at higher risks, like smokers, before symptoms even appear.
Yeah, so adding on, the reason why this test is advantageous to other tests is because it's minimally invasive, which means that it doesn't require anything to be taken out of your body. For example, scientists have previously used biopsies to test for cancer, and a biopsy is basically an invasive medical procedure to remove cells, tissue, or fluid for a microscopic examination to diagnose for cancer or other diseases. And these can be painful and cost money and take time to get your results back. So this is a really much more advantageous way to test if you have cancer. So building onto what Ellen said before, she mentioned that the hydrogel is able to capture proteins, and the reason why it's able to do this is because it's a network of polymer chains that can absorb and retain large amounts of water, and it's also used in a bunch of different things because it has such unique qualities. It's not a solid or a liquid, and it behaves like living tissue. And then when this gel is exposed to light, like she said, the radiation, it acts as an external trigger that causes the polymer network, which is what the hydrogel is made of, to break down or change its structure, and then the trapped molecules the proteins can escape, and then you can detect for those different proteins and see if the person has cancer or not. Did you want to add something, Sharvi?
Yeah, so I wanted just to talk about how you were talking about the polymer chain and stuff. Also, the hydrogel has different pore sizes in it, which allows for protein diffusion. And then it's chemically modified also to bind to specific cancer-associated proteins. And then it also reduces background noise from different irrelevant saliva proteins so that the hydrogel can specifically capture the proteins released by the cancer cells. And then there's also, once sampling is complete, like Shanti and Ellen were mentioning, the hydrogel is exposed to UV light. And basically what happens is that the UV light breaks the photocleavage chemicals, the photocleavable chemical linkers, which are in the gel, and that basically releases the captured proteins all at once. And then this basically means that the release is controlled and it's also reproducible. So like you know when you can release the proteins, and then the proteins also stay intact, and detection becomes more accurate and more sensitive. And then the released proteins are then analyzed using this thing called fluorescent labeling, where signal intensity kind of correlates with biomarker concentration. So because the biomarkers were pre-concentrated in the hydrogel, the system can detect much lower protein levels than standard saliva tests, which basically enables earlier detection. So basically the Lollipop is like a sampling device, and it's also a biochemical concentrator. So instead of trying to detect tiny signals in like messy saliva, it engineers the sample itself so that it makes cancer detection easier and more reliable. Serena, do you want to talk about the pros and cons?
Yeah, sure. So, some of the pros would be that, as Shanti said, they're non-invasive compared to blood tests, biopsies, or imaging, since it relies on saliva collection. It could help with earlier detection of certain cancers, which is often linked to better outcomes. It is easier and more accessible than many current screening methods, and potentially increasing the participation. And they're also saliva-based tests are generally cheaper and faster to process once the proper technology is established and mass-produced. And it may be useful for repeated screening or monitoring since it is low risk and easy to perform. For some cons, there are risks of false positives and false negatives, which could lead to unnecessary stress and follow-up testing, or delay diagnosis and treatment, which is bad, obviously. Not all cancers release detectable biomarkers into saliva, which limits what it can realistically detect. Saliva composition may be affected by diet, oral health, infections, medications, and smoking, so it might not be completely accurate. And ethical and regulatory concerns exist around accuracy standards, medical oversight, and data privacy. Anyways, that wraps up for today's episode. I'm Serena.
I'm Sharvi.
I'm Shanti.
I'm Ellen. Just a disclaimer, this podcast is made, researched, and created purely by high schoolers, so everything we say is not 100% factual. Thank you for listening.